In 1917, a food chemist named James Currie made a promising discovery. Any strain of the filamentous mold, Aspergillus Niger, would produce high concentrations of citric acid when grown in sugar medium. This tricarboxylic acid, which we now know is an intermediate of the Krebs cycle, had previously been extracted from citrus fruits for applications in food and beverage production. 2 years after Currie's discovery, industrial-level production using Aspergillus Niger began, the biochemical fermentation industry started to flourish, and industrial biotechnology was born. A century later, citric acid production using this mold is a multi-billion-dollar industry, with Aspergillus Niger additionally producing a diverse range of proteins, enzymes, and secondary metabolites. In contrast to what most people might think, citric acid is not, or not anymore, isolated from citrus fruits, but is industrially produced by the filamentous fungus Aspergillus Niger. The process was pioneered by James Currie, a food chemist, who 100 years ago published a study describing the superior properties of Aspergillus Niger for the industrial production of the acid. In particular, Currie demonstrated the necessary growth medium for citric acid biosynthesis and the fungus's ability to grow at low pH levels, 2.5 to 3.5, while still producing high amounts of the metabolite. Moreover, this work demonstrated the direct correlation between the amount of substrate in the medium and the amount of product, laying the basis for modern-day industrial fermentation of citric acid. In contrast to other species of fungi that had been reported to produce citric acid by 1917, every single strain of Aspergillus Niger that Currie tested could efficiently produce this molecule when grown in sugar solutions. 2 years later, the American company Pfizer made a pilot plant for biochemical production of citric acid, and by the mid-1920s, production using Aspergillus Niger fermentation far outweighed extraction from citrus fruits. The citric acid cycle was comprehensively determined over the next several decades, resulting in the award of the Nobel Prize to Hans Krebs and Fritz Lippmann in 1953. The first and final reaction in the cycle involves the formation of citrate from oxaloacetate, acetyl-CoA, and water by citrate synthase, ultimately generating chemical energy in the form of adenosine triphosphate, ATP. The objective of industrial microbiologists, including James Currie, was to exploit this cycle, and indeed many other metabolic pathways, to ferment useful molecules. Although there are variations in fermentation techniques, in general, industrial production of citric acid requires aerobic, submerged growth of Aspergillus Niger in a sugar solution, which is usually derived from inexpensive sources such as molasses, corn steep liquor, or hydrolyzed corn starch, amongst others. After fermentation, Aspergillus Niger is physically removed, usually by filtration, and citric acid is isolated by precipitation of the fermentation mix with calcium hydroxide, lime, to generate calcium citrate salt. Subsequent treatment with sulfuric acid yields the citric acid product. The widespread applications of citric acid are shown by current figures regarding this metabolite. In 2007, worldwide production was estimated at 1.6 million tons, with an estimated value of $2.6 billion in 2014 and predicted to rise to $3.6 billion by 2021. As a weak acid, it can be used as an antioxidant, preservative, acidulant, pH regulator, or flavor in food and beverages, as well as comparable applications in the pharmaceutical and cosmetics industries. Citric acid is currently predominantly produced in China, which accounts for approximately 60% of global production. However, Aspergillus Niger industrial applications are not just limited to the production of citric acid. As a prolific secretor, Numerous industrially relevant enzymes and other molecules are produced by this fungus. Analysis of 100 years of Aspergillus Niger publications indicated that citric acid and fermentation were amongst the most common returned words in titles from every period. Clearly, Currie's discovery of conditions for maximizing citric acid production was indeed a biotechnological revolution, and one that would constitute a major focus of research over the next century. Given the tremendous advances in the knowledge of Aspergillus Niger biology over the last century, and the concomitant development of bioinformatics, cultivation, and molecular tools which are now at the disposal of the community, this industrial fungus has the potential to remain one of the most versatile fungal platform microorganisms. Aspergillus niger offers a chassis for products which cannot be produced in easier-to-handle bacterial systems, and it can produce not only proteins and enzymes at high concentrations, but also pharmaceuticals, which are beneficial for human and animal health. It is predicted that Aspergillus niger will be one of the key organisms involved in the next industrial revolution, the change from a fossil-based economy to a bioeconomy.